to my wonderful champions. Let me just wait for a second and see if the network is fine, if I'm visible and audible. A quick second there. Okay. Hi Manjunath. How are you, dear? I hope you are doing great. Well, let's wait for a minute's time so that everyone else who wants to join come in as well. So today we are going to solve a very important chapter, that is molecular base of inheritance. We're going to solve questions related to that. And first of all, my dear children, so we have shifted the timing to 6 p.m. in a belief that you guys would be able to attend the live sessions even well, because some of you we saw that were not able to attend the live sessions as your classes ended at so point of time. So my dear children, we have shifted the timings. I hope it helps, and one promise that you have to give to us is that you would be regular from today onwards. Okay, so this chapter gives you 5 questions every single year. 5 questions means what? 5 marks in your KCET examination. If you study it properly, basically you are going to score 5 more marks than your competitor out there. So your head should work like that, and as I said, we'll wait for a quick 1 minute so that everyone else gets to join. I see 7 people. So all my dear children who are watching this video, please try to engage over here. Please give me the answers before I do. Okay, I love talking to you children. Okay, I love engaging with you guys. So make sure you are making this live super strong and super fun. Okay? So wishing you the best, and starting ahead with the first question. Those of you who know the answer for this question, tell me 1 A, B, C, D. So it goes like this. To find the sequence of binding of the following amino acyl tRNA complexes during translation to the mRNA transcribed by the DNA segment having the base sequences this. So those of you who read this question for the first time basically are like, oh my god, okay, so this is the tRNA that I have, and basically this probably is the mRNA, and I would have to just find what's the consecutive tRNA opposite to it. That is the thing. So Manjunath says the answer for this question is 1B. Anyone else who is attending this live thinks that he's wrong? Well, first of all, that you need to know about this question is that never in an mRNA you will see T. Thymine is not there in RNA, it's there in what? DNA. So basically over here they strictly mentioned that this is a DNA strand. So first of all, you have to convert this DNA strand into what? mRNA. You have to make it into an mRNA, and the direction of mRNA will be from 5' to 3'. So when you do this, so T will be base pairing with what? A. T will be base pairing with A will be base pairing with what? U this time, and C will be base pairing with G, and this is how it will go. A, U, G, and the sequence goes on. So first of all, we have to make an mRNA, because the mRNA will now be translated with the help of tRNA. Okay, so how does it go? How does it look? Look over here. So over here, you have this as a template strand. Now you make an anti-parallel strand for that. So whenever you make, please make sure you're underlining 3 codons out there. Otherwise you might make mistakes. So TAC it was there, so now it will be AUG, and the same goes for all of them. So this is what your mRNA, which is in 5' to 3' direction. Now translation occurs in this direction only. So AUG, if it is there, then the tRNA, there will be something opposite to it. So if it was AUG over here, my dear children, in the anticodon, what will you have? You have U, A and C. So this will be the first one out there. It will be UAC. So in that case, UAC over here is B sequence. Over here is B sequence. Well, you do not have to do this much drama. It will be so easy. You do not have to do all of this thing. Just replace the T by U. Just replace wherever T is there, please replace by U, and you will get the whole thing. You will get the actual tRNA codons which will be there, and with that you will be able to. So over here it is what? UAC, again AUG, and then GG U and then what? GGU and then CC. It was G over here, so it remains to be the same. This is how it will be going. This is the pattern. Fine, so B, A, D, C would be your answer. First B, then A, then. So Manjunath, you were absolutely correct. Kudos to you. Now let's go ahead with the next question out there. This is related to semi-conservative replication. Now those of you who know the answer for this question, be quick and tell me the answer, okay? So E. coli bacteria grew in NH4Cl, where the nitrogen isotope was 15, okay, medium. For several generations are allowed to grow in NH4Cl medium, but over here the isotope is 14. So after 2 generations, the bacteria are isolated from the medium and the DNA of the bacteria centrifuged in cesium chloride, CaCl. The result of the density gradient of DNA is what? So first of all, we had E. coli which were being grown in this medium. So that means the DNA strand had nitrogen which was having N15 itself. Now what did you do? Basically, you shifted it to another medium which was having N14 isotope. Now they are asking you after 2 generations, what are you going to find? After 2 generations, the bacteria isolated from the medium, and what are you going to find? So Manjunath is saying the answer for this question is 2D. Is it this? Is it 2D? Let's find that out. So over here, when this DNA is going to segregate, my dear children, so over here you have one strand, here also you have one strand. These 2 strands are what? Of N14. These 2 strands are the new strands that are being formed in response to the older strands which were there. So replication happened, this replicated this also replicated to that, and over here, so that means one strand will be N15, another will be N14. Similarly here also, one strand will be N15, another will be N14. This is first generation. You are asked about second generation, 2 generations. So again, my dear children, it is grown in N14 medium. Now what will you find? These 2 strands which are there, they will again move apart from each other. So now this time I am going to draw like that. So this is one strand, okay, and this is another strand. The same thing applies over here. There will be one strand over here and again and yellow strand. So this is N14, N14, here also N14, N14, and this will be what? N15. Here also it will be N15. Now the new strand of the DNA which will be accompanying them, my dear children, will be what? Will be N14 type only, because we are growing in the same medium. So this is, it looks like this. So what do you find? Over here you have 2 strands which is just made up of N14, N14, and we have 2 strands which are hybrid. That means it is made up of N15 as well as N14. So what do you find? Both hybrid and the light DNA. So you call this DNA which is of N14, my dear children, as light DNA because it is a lighter isotope. Fine, in the density gradient medium, it settles. So 3 things you can see usually. So when we started off, they were settling somewhere over here because their density was super high. Now when you got this, their density was less in comparison to this. So hybrid density is less. Now the most lesser density will be of N14, N14. So you will find light DNA and also hybrid DNA. So answer for this question would be D. Manjunath, you are correct again. Superb. Now we will go ahead. So the same thing is being mentioned over here. If at all you want the PPT, you will get it. Make sure you are following us on WhatsApp. Okay. So question number 3. Read the following statements carefully and choose the correct statement. In a transcription unit, the promoter is located at the 5' end of the coding strand. You have to find the correct statement. Let's see who is the one who says the answer first. Okay, so in a transcription unit, the promoter is located at the 5' end of the coding strand. That is the first statement. Next, the single strand DNA having the polarity 5' to 3' is the template strand. Do you think it's a template strand? Next, RNA polymerase binds to the operator during transcription. Does it bind to the operator during transcription? And also, single base DNA differences occur in humans and are known as single nucleotide polymorphism. We'll go one by one. First of all, in a transcription unit, the promoter is located at the 5' under the coding strand. So my dear children, you have 2 strands. One is the template strand and the other one is the coding strand. The coding strand is always in 5' to 3' direction, whereas template strand will be in 3' to 5'. Why? Because template is the one that undergoes the process of transcription. Now you are being said that the promoter is located at the 5' end of the coding strand. That means somewhere over here. Yes. So ma'am, why not 3' under the template strand? You do not refer to it like that. You always refer coding strand and say. So yes, this is correct. The promoter is located over here. So I'll say statement A is definitely correct. Now statement B, the single strand DNA having the polarity 5' to 3' is the template strand? No, it is the coding strand. So this statement will be what? Wrong. Okay, next statement, RNA polymerase binds to the operator during transcription. So now there is a region known as operator region. Do you think RNA polymerase, which is responsible for the transcription of this template strand, it binds on the operator region? No, it actually binds on the promoter region, and from when it attaches over here, then only it will be able to initiate the whole process. So this is again wrong statement. Now last statement, single base DNA differences occur in humans are known as single nucleotide polymorphism. Yes. So basically suppose I have AGCT. Over here you have ACGT. So this is a single base that is being changed from G to C, and we have it, and this is known as single nucleotide polymorphism. So A and D statements are the correct one. Anjanath said the answer is 3D. Let me see. Yes, the answer is 3D, and my dear child, you are absolutely correct. What about the others who are watching this video and not giving the answers? Come on children, aren't we preparing for KCET examination? Aren't we preparing for KCET examination? In that case, we have to try. You do not need to be worried about will you get the answers wrong or right. Listen, we are here to study. There are times that we might get answers wrong. And this is how we'll remember better. Okay, I got it wrong there. Now I remember it. Now I'll not make the mistake. So please do not move away from not attempting the question. Try to do the question. Okay? Chalo. Next question. Try this out, and this will be only done by the children who have read the NCERT really well. So it's about the DNA content. Match the number of nucleotides of genome of column 1 with the organisms of column 2. Choose the correct option given below. So over here you have all the option. You have to choose what is right. So 5386 nucleotides, in which organism do you find 5386 nucleotides only? 4850, you have so many options. I'll tell you one easy thing. It's always easy to look for human beings' DNA. It can be either 6.6 into 10 raised to the power 9, that is the diploid content, or half of it, that is 3.3 into 10 to the power 9, will be the haploid content. That means the half content that we have in the gametes and all. So I can say D goes with Q. Do I have an option like that? Ma'am, I have one option, and in that I have one option, and basically with that I can find the answer. Prajwal, basically those of you, anyone probably would watch it later on and it would be beneficial for them, and that's why I'm teaching. Okay, so Manjunath, yes, right over here says 4C is the answer. You are absolutely correct. So my dear children, you should, suppose over here you do not have an option like this, like man, it's not easy option that you get. So what do you have to do? You have to look for the other things. You need to study that. So E. coli is the next option that you can remember easily. The whole content of E. coli is 4.6 into 10 to raised to the power 6 base pairs. This is again an easier thing. Okay, now 48502B is found in T. So over here, bacteriophage lambda, the one that infects the E. coli, has this much amount. And now we are left with this guy. It goes with 5174 bacteriophage, another type of bacteriophage. So as I said, if you know these two things, probably you might be able to decode the whole answer. So look for the elimination method, eliminate whatever you are sure of, and then go ahead with these things. Cool? Let's go ahead. The next thing that we have again has something to do with semi-conservative DNA replication. So my dear children, you know what? We have actually launched a PYQ book. So as of now, it's available for physics, chemistry, as well as mathematics. So if at all you are someone who generally wants to practice more PYQ questions, more than what's given over here, we are solving 20 questions. And if at all you want to have more practice sessions, please make sure you are getting this book. The link is given in the chat sessions itself. Okay? Now, as I said, we are going ahead with this question. Those of you who know the answer for this question, comment it as soon as possible. And Manjunath, you are going super awesome. So if E. coli is allowed to grow for 40 minutes in a medium containing N15, then the number of N14 containing DNA would be, this is super easy, I'm telling you. So E. coli is there, it is grown for 40 minutes. You know, in every 20 minutes, E. coli basically divide. That means in every 20 minutes, DNA replication is happening. Now this E. coli has been grown in which medium? N15 medium. And now what are you being asked? You are asked the number of N14 containing DNA. That means a DNA, that means DNA containing what? N14. How can a DNA contain N14 if at all it is not grown in a medium containing N14 isotope, my dear children? This is just to make you confused. So your answer for this question would be A0. Manjunath, super. What about the other children? Come on, dear children, come on. Let's try it out. Okay, those of you who are watching this video who are shy of not answering the questions because you feel that you might get things wrong, please trust me, this is how you'll remember things better. Okay, so your answer for this question is 0. Do not go by these numbers. This is just to fool you guys. Now, again, it has something to do with the semi-conservative DNA replication or generally DNA replication as a whole. Okay, this is just for DNA replication. Polymerization of DNA nucleotides during synthesis of lagging strand occurs in which direction? So they are asking you the DNA replication is happening in which direction? And to be specific, they are talking about the lagging strand. So we all know, my dear children, in DNA there are two strands. Okay, in DNA we have two strands. One strand is in basically 3' to 5' direction, and the other strand which is there, it will be in 5' through 3' direction. So DNA polymerase, which does this function, which does DNA replication, it basically will prefer this strand and will start making the DNA in 5' to 3' direction. So the other strand which is there where things are not that smooth, you call that strand as lagging strand. And now they are asking you, in which direction does DNA replication happen in lagging strand? This is just to confuse you again. There is only one direction in which DNA polymerase work. There is only one direction in which DNA replication happens, and that is just 5' to 3'. Always, whether it's lagging strand, whether it's leading strand. Yes, Manjunath, the answer would be what? B. It would be B. Cool. And children, if at all you're watching this, share this with your friends. Listen, it will be more engaging if at all you also try it out. Otherwise, it is going to be a super boring lecture. Okay? So let's be friends and let's try to give the answers for the question. Okay, question number 7. Which of the following sequences of mRNA are required for translation process but are not translated? Any idea? So this is an mRNA, okay? It's an mRNA, and I'm telling you, there are some sequences in the mRNA which is very much important for the process of translation to happen. That means for tRNA to come and bind and for a protein to be made. For all of that, you need this particular sequences, but they are not going to get translated. They are not going to form any amino acid. They are not going to code for anything. Any guesses what is the answer for this question? This is 7th question. Give me the answer right. Stop codons, anticodons, sense codons, UTR. What is the answer for this question? And over here it is asked. It is very much important for the translation process. Anyone who is watching this video, do you know the answer? So I will not tell the answer until you try it by yourself. Come on. Super important question. Stop codons basically are responsible for stopping the process of translation, right? It is not required for initiating the process of translation. Anticodons are there in the tRNA. Again, it's not in the mRNA. Sense codons, my dear children. Again, no. What is the thing that is there? Any guesses? Come on, come on, come on. 7D. Manjunath says it's D option. And Manjunath is absolutely correct. It is UTR. You know what is the meaning of UTR? What is the full form of UTR? It is known as untranslated region. That means it is not going to undergo translation. And where do you find it? This is again a super important question. You get it in the form of theory questions also in your PU2 boards. So over here it is found before the 5' end and also after the 3' end, and it is super important for the process of translation to happen. So your answer for this question is absolutely correct, Manjunath, and those of you who didn't know that, I hope now you know the answer. So this is the UTR, 580 UTR and 3' UTR. Cool. Let's go ahead with the next question. And dear children, I have chosen those questions which are mostly repeated every single time, so that you get a full coverage of whatever you are doing. Okay, so DNA present in the nucleus was named as nuclein by, so one day one random guy came and said that I am not going to say it as a nucleus, rather I'll give it a different name, we'll call it nuclein. Who was that particular person? James Watson and Crick, Frederick Misher, Maurice Wilkins, Rosalind Franklin. Any guesses? This is in the 3rd page of your chapter. Okay, it's in the 3rd page of your chapter. Names of all the people who are. Aishwarya is here. Hi, dear. So Aishwarya Kamath is back. I'm super happy to see you over here. You are saying the answer for this question is 8B, Frederick Misher. Is she correct? She is absolutely correct, and I want more of you guys to come and attempt this. So these are the things that you need to remember. First of all, 1869, long back, Frederick Misher came. He was a Swiss physician. He said that some acidic substance was there in the cell, okay, and he called it nuclein. First guy. And now came Watson and Crick, my dear children. They were able to deduce the structure of the DNA. That is James Watson and Francis Crick. Then, my dear children, they used the information from Maurice Wilkins and Rosalind Franklin. So these two people who were there, they were working on this X-ray and X-ray diffraction method, lot of things. And with the help of this data, Watson and Crick were able to deduce the whole structure. Okay, were able to deduce the whole structure. Now, base pairing such as A will be pairing with T and G will be pairing with C and all of that was told by the next person, whose name was Erwin Chargaff. Okay, his name was Erwin Chargaff. So these are the names that you definitely need to know. So your answer for this question is this. Aishwarya, sorry ma'am, it's totally fine, dear. Totally fine. I am glad that you joined. That's more than enough. Okay, chalo. Next question that we have over here has something to do with the DNA fingerprinting. Manish Sirpur, helpful. Thank you. Okay, Manjunath also says the answer is 8B. Super good. Okay, so in which of the following steps in DNA fingerprinting technique are labeled VNTR probes used? This is again a question related to DNA fingerprinting, super important concepts. So they are asking you, in which step of DNA fingerprinting do you use this VNTR probes, variable number of tandem repeats probe? So it's just nothing, you can remember it like that. It's a sequence of DNA which is labeled by a radioactive compound. It is labeled with a radioactive compound such that my dear children, suppose I have the DNA and I put this probe. Okay, I put this probe over here. This probe will go and bind with only that particular DNA which has the complementary base pairs. Suppose it binded over there, here. Now, if I want to see this, what do I do? If I want to see this, basically I have to make sure that I put some rays, put it under some UV rays. In that case, under the UV rays, they will glow up and I will be able to see the entire sequence where it is going and matching. Okay, so basically, can I say that during the isolation of DNA, do you require it? No. So Aishwarya has told the answer, it is 9D. So this process of basically, what are you doing over here? You are having the DNA and the probe. This is a hybridization method that is going on. And also, my dear children, all of this, the DNA segments are separated using a machine that is known as electrophoresis. Okay, not machine, a method that is known as electrophoresis. So there it is not visible. The DNA strand won't be visible. You take a nitrocellulose sheath and you put it on the, electrophoresis chamber. You transfer all the DNA over here in this particular sheath. This is known as southern hybridization or blotting method. This is known as blotting, and this is where you will be actually seeing the DNA which is matching. So it is during hybridization. You are absolutely correct, Aishwarya, as well as Manjuna. The answer for this question would be D, during hybridization. So this is how it goes. You can go through it properly if at all you download the PDF once. Cool. Next question that we have is related to Human Genome Project. So according to Human Genome Project, the total number of genes in human genome is estimated at 30,000. The number of genes present on the Y chromosomes are. So a total number of genes which is there, it is what? 30,000. And now you are asked, what is the number of genes present on the Y chromosome? You know, Y chromosome is the tiniest chromosome that is there, and it is found in males. Guys have XY chromosomes, right? Guys have XY chromosome. So Y has the least number of genes. Always remember, it is the smallest and it has the least number of genes. So what will be your answer for this question? What is the least number over here? We are doing 10th question and I am asking you, what is the least number of genes present over here? Any guesses? 2968 genes, 242 genes. Manjunath says the answer for this question is 10C. Yes, it is 10C. 231 genes are present on the Y chromosome. Only 231 genes. And do you know, the highest number of genes are discovered in chromosome number 1. Chromosome number 1 has the highest number of genes being discovered. Can you tell me what is that number? Anyone out there? It is there in the option. It is there in the option. That is why I am asking you. In chromosome number 1, you have the highest number of genes. Any guesses? So this way we are going to study. We are studying two things at once. Come on, come on, come on. If no guesses, then I'll tell. But if yes guesses, then you have to tell. So 231 in Y chromosome, and in chromosome number 1, you have 2968 genes, the highest number of genes. Always remember, you may get an MCQ question also in your PU2 boards. So your PU2 boards are also going to come. So this preparation is going to help that preparation also. Remember, guess Aishwarya, it is 2968. Okay, cool. Next, it is related to DNA fingerprinting again. Try this question. It's a very fun question. So in a crime investigation, the investigator or investigating officer collects different biological samples from the crime spot for DNA fingerprinting analysis. Which are the following samples is not helpful in this analysis? That is the question. So you know you are in a crime spot. Some murder or something has happened, okay? And there are so many samples. There are hair follicles, okay, there are skin shreds, there is your semen sample, there is erythrocytes, lots of things are there. But which of them do you think is not going to be helpful for the forensic team for finding out who is the actual killer? Any guesses? Any guesses? What is the answer for this question? Is it our skin? Is it the semen? Is it the hair follicle? Is it the erythrocyte? What is the answer? Come on, come on, come on. 11th question. Manjunath says 11th answer is B. Why B? Any idea why B? Aishwarya also says it's B. Okay. Why do you guys say it is B? What is erythrocytes first of all? Erythrocytes is your RBC, red blood cell. So your hair, your skin, the semen, everything, my dear children, over here, just a second, okay? Yeah. Hair, skin and the semen would be having the DNA content, but RBC does not have, because RBC does not have the nucleus. It does not have the nucleus. So initially RBCs have the nucleus, but since RBC have a very huge responsibility of carrying oxygen, it will prefer to give that space to hemoglobin molecule, which is why when the RBCs mature, they kick out the nucleus. So they do not have the nucleus, which is why they stay alive only for 120 days. That is the reason. If nucleus is not there, do you think DNA can be there? If DNA is not there, can I do DNA fingerprinting? No. So your answer for this question would become B. So do not mug your answers up. Always know the reasons of why an answer is wrong or right. Now the next question is a 12th question. This is related to RNA processing. Okay, so in eukaryotes, the entire base sequence of a gene does not appear in mature RNA because... So in eukaryotes, the entire base sequence of a gene does not appear in the mature RNA. Means what? Basically whatever is there in the DNA, over here it is not found in the mRNA. Why is that so? Any idea? This is 12th question. Try it out, my dear children. So all the sequences which is there in the DNA or which is there in the heterogeneous nuclear RNA is not found in the mRNA. You are being asked the reason for that. This is 12th question. Your options are: some gene sequences are removed by exonucleases. There are some enzymes which remove some gene sequences, which is why you do not see it over here. Is that the reason? Or transcription in eukaryotes consumes more energy. That has nothing to do with this question, right? Manjuna said they do not contain any genetic material. Yes, yes, my dear children. Okay. Fine. So Aishwarya says the answer for this question is D. Introns are removed during processing. It's that so? So let's see the next option. Coding sequences are removed during processing. That is also not there. Why do you remove coding sequences? Coding sequences mean the sequences that will actually code for proteins. Will you remove it? No. What do you call the sequences as? You call this as exons. My dear children, there are non-coding sequences over here which you call as introns, and which is why Aishwarya is super correct. So introns are removed during the process of processing. We do not need introns. Why will we need that? It does not code for any amino acid. So I will just remove it, and that's how I'll get my mature mRNA. Okay, yes Manjunath, you're also super correct. So your answer for this question is D. Cool. Next question okay, I'll show you this. And this process, what do you call this process as? You call this process as splicing. Always remember. So over here, okay, I have the exons, and in between I have the introns. I remove the introns, as you can see, and now I have the mature mRNA. Fine. Next question, question number 13. When Escherichia coli, it's not coil, okay, it's coli, Escherichia coli, cells are cultured in a medium where lactose is absent, the I gene of lac operon continues to produce repressor mRNA because, why is it so? Why does it continue to produce the repressor mRNA? So it is being asking. So this is the lac operon. This is how lac operon looks. Over here you have a promoter region for a gene which is known as I gene. Okay, now you have another promoter region and you have an operator region, and there are structural genes over here, my dear children. You call them Z, Y, A. Each of them are going to code for some enzymes. Okay, they're going to code for some enzymes. So usually what happens, this I gene which is there, it codes for a protein which is known as repressor protein. And you know what it does? It is very dusht in nature. Repressor protein, it goes and binds on the operator region of the next gene. And when it does, do you think the RNA polymerase which is going to come bind over here can do the transcription? No, it won't be able to move ahead. So the whole transcription is stopped right there. Now your question is, when I am culturing the E. coli has this, okay? And when I am culturing this E. coli in a medium where lactose is not there, okay, when lactose is not there, the I gene of lac operon continues to produce the repressor. So usually when lactose is there, you know what happens? The lactose goes and binds on the repressor protein. So it won't be able to go and bind on the operator region, and the whole process happens smoothly. When lactose is not there, this guy, so when lactose is there, this guy stays over here. So it has been asked, why do you think the I gene is basically continuously producing the repressor protein? That is the question. Any guesses what is the answer for this question? Now that is kind of different and kind of a tough question for some of you probably who might not have practiced these type of questions earlier. Is it because I gene is a structural gene? No, my dear children. A gene is known as structural gene when it has anything to do with the structure, whether it makes some proteins or enzymes which give some structure or hampers the structure or has anything to do with the structure. Is it because it's a non-coding gene? If it was a non-coding gene, it won't be producing repressor protein. Is it because it is an operator gene? It is not an operator gene. What is the reason for this? Manjunath, I think, gave me the answer for this question. It is 13D. It's a constitutive gene. Okay, it's a constitutive gene. That means a gene which continuously produces what? The protein. It continuously undergoes transcription and produces the protein, which you call as the constitutive gene. Okay, we want the lac operon to be closed when lactose is not there, which is why it is continuous supply is there. So your answer for this question will be D. Yes, Aishwarya. I hope this is clear and I hope you will never forget it. Okay, so this is how it looks. So this is the regulatory gene which is there, which is going to produce the protein, repressor protein, and rest all of them is right over here. The next set of gene is right over here. Next question, question number 14, and the topic over here has something to do with translation. Let's try it out. In which region of the tRNA molecule is the amino acid binding site located? So you know tRNA, it looks like this. Sorry, okay. tRNA, it almost looks like this. So there is 5' end, there is 3' end, and over here you have an anti-codon loop. That means it will be having an opposite codon of the mRNA, and that is how it comes bind over here. So for example, AUG is there in the mRNA. Over here it will be UAC. Okay, so they are asking you, where does the amino acid come and bind? So Aishwarya is saying the answer is 3A. Sorry, C for this question. Is it C? Does the amino acid come and bind over here? Well, the answer is yes. It is at the 3' position that the amino acid will come and bind. So basically amino acid will be there, you know the anticodon is there. Now this guy will be going and binding over here. That means we have to produce this, we have to give this amino acid, which is methionine. The same thing goes again and again in the whole translation process. So always remember it's 3' end. And do you know the name of the process? What do you call this process as? Where amino acid is added? Anyone who can tell me the name of the process where the amino acid is added? Any guesses? The name of the process when amino acid is added? Anyone? You need a high amount of ATP, okay? This is high energy requiring process. You need high amount of ATP, and you need to know the name of the process. It is known as charging of tRNA. It is known as charging of tRNA, and also you have one more name that is known as amino acylation. The next name of it is what? Amino acylation. Cool? Chalo. Next question. So this is how it looks. So you would be having your anticodon loop, and there is 3' end, there is 5' end. On the 3' end, you have the amino acid being joined. Cool? Chalo. 15th question, it has something to do with semi-conservative replication. E. coli fully labeled with N15, okay, E. coli which is fully labeled with N15 is allowed to grow in N14 medium. The two strands of DNA molecule of the first generation bacteria have what? Have what? So usually we initially started off with two strands having N15, N15. Why N15, ma'am? Because it was grown in the medium which had N15. Now what are we gonna do? We are going to grow it in a medium that has N14 isotope. In that case, usually, my dear children, what happens? When E. coli is dividing, also the DNA content would be dividing. The DNA content which is there, it also divides. So over here, you have two strands which get separated, both of them N15, N15, and the new strands which will be forming, it will be taking this nitrogen. So which is why I'll say it is N14, N14. Now you know the difference of these strands and this strand? Okay. Ma'am, sorry for the disturbance. Please can you tell this PYQ notes, because I checked Sankalp series, only one is there is now series. Okay, so we'll definitely put up the PDFs. Do not be worried, Fatima. Okay. We'll make sure we put up it really soon. If at all it's not there for the other. For mine, it has, this video has to end, then only I can upload. But yes, we definitely check through it. Okay? So over here, yeah. So basically, if at all I have cesium chloride medium and I try to separate this. So you know, it separates down here because it has high density. So N15 isotope has high density, because which is why the DNA puts over here. But now if I try to centrifuge this guys, here they will be segregating a bit top, because they are comparatively lighter than the N15 ones, and you call them hybrid. Why do you call them hybrid? Because you have both N15 and N14. So your answer for this question becomes what? So I have Aishwarya who says the answer is B. Okay, Manjunath who says the answer is C, and Sanika Patil is there. Hi dear. So you are also saying the answer is C. Well, different density but do not resemble with the parent DNA. This is the answer. So they are asking you the two strands of DNA molecule of the first generation bacteria, doesn't it have different density? Yes. This is what? Having a hybrid density over here, it is lesser. And also, does it resemble with their parents? No, they look different, right? They are made up of different things, so they look different. So your answer for this question is actually C. So kudos to those of you who tried giving the answer. You are doing super great. Okay? Rupa, hi ma'am, good evening. Hi dear, very good evening. Fatima, tomorrow class is not there. Okay, so PYQ live session is not there since it's Saturday. We'll be coming back live on Monday. And by the way, for all of you who are watching this video, we are bringing up a series for PU2 and it's going to be super good series. I hope all of you know that in KCET 50% of weightage is given to your PU2 score, so make sure you're not messing it up. Whenever you're studying for some competitive examination, have PU2 at the back of your head. Definitely solve questions from your PU2 question papers as well. Okay? And we are going to launch it soon. On Monday, you will have the first class. Be there. Cool. Now, next question that we have is this. Now, those of you who have read your NCERT really well, only those of them would be able to answer this. Experiments involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed on which plant? So, basically, it is in the semi-conservative replication mode of DNA. That part of the chapter itself, they are talking about, you did an experiment where you used radioactive thymidine instead of using NH4Cl, okay, which had N15 and N14, you use radioactive thymidine. So in which plant was it done? So I see a lot of new people out there. I missed you guys in the initial part of the video. But now we are here and I'm super happy. So yes, 16A, Rupa says A, okay. Prachi also says A, Aishwarya also says, that means 3 of these girls have read the NCERT, super good. Yes, the answer for this question is Vishya faba, or also you call this as faba beans. Okay. Yes, Manjunath. Super good, dear. Fine, do not forget there are small details in your NCERT which you should refer to in between, okay? Do not leave your NCERT like that. Now, the number of codons effective in coding 20 amino acids are what? Tell me the answer. Hi, Sanika, your answer was also super correct. Now, try solving this question, 17th question. Answer me in 17A, B, C, D format. Okay, the number of codons effective in coding 20 amino acids are. So we know there are 20 amino acids. Aishwarya says the answer for this question is B. Is it B or is it A? Sanika also says it's B. Very good. All of them very clever. Super good. Why is it not A? Why is it not A, my dear children? Because you have been asked about the ones that code for amino acid. Remember, even though we have 64 codons, okay, even though we have 64 codons, 3 out of them are going to code for what? Are not going to code for any amino acids. They are stop codons. So the ones that actually code is just 61, which is why your answer would be B. Okay. Yes, stop codon. Yes, Rupa. So all of you, I think, yes, Sanika, Rupa, Aishwarya, and Manjunath is correct. Pachi, let's see. Let's check the answer once more. I hope you're trying the 17th itself. Okay, so I hope you got the answer for this question, dear children. Now we'll move ahead with the next question, 18th question. Which of the following statements are correct with reference to prokaryotic genome? Again, those of you who have read the NCERT well will be able to answer this. You are asked what is correct. So whenever these type of questions come, no, first of all, underline what are you being asked. Correct or incorrect? Correct. And what are you being asked about prokaryotic genome? That means the genome of bacteria and all of that. Prokaryotes, you know, they do not have any segregation of nucleus, cytoplasm, anything, because they do not have the nuclear membrane, the what do you call organelles which has the membrane. All of it is not there in prokaryotes. So you have to tell what is perfect fit for prokaryotes. Anyone? 18th. Question number 18th, A, B, C, D. So all of the 12 children who are watching this video, I want 12 answers from you guys. Come on, you can do it. You are super awesome kids. Do you know that? And from the next live onwards, try to be there at 6. For you guys, we have shifted the timing. We are staying late. Okay. So make sure you are over here with us sharp at 6 o'clock. Chalo, to this question. I'll move aside. What is true for prokaryotic genome? Monocistronic structural genes. Do you think it's monocistronic prokaryotes? Monocistron means what? Basically, over here, this is a cistron which is having a gene coding for only one particular protein. Okay, a gene will be coding for one particular protein. Again, you can have separate gene coding for another one. But over here, only one gene. Polycistronic means basically this is a cistron having 3 genes probably, this coding for one thing, this coding for another protein, this coding for another protein. That is known as polycistronic. Okay, so where do you find polycistronic, my dear children? You find polycistronic in the case of prokaryotes. In the case of eukaryotes, like us, we find monocistronic. We are more organized. Okay, now, so Bhagyashree is there. Prachi is saying the answer for this question is B. Only B, C and E. Okay, let me see. And Bhagyashree also says that. And Rupa and Sanika are talking about, yes, polycistronic. You guys are correct. Fine. So this particular statement is wrong. We got to know that. Now, introns absent in the structural genes. What are introns, my dear children? I told you, basically, in the normal structure of the DNA or in the hnRNA, you have regions like exons which codes for amino acid, and there are regions known as introns which do not code for any amino acid, basically proteins. So these introns are being removed, okay, are being removed to form the mRNA, to form the mature form of RNA. Now, in the case of prokaryotes, they do not actually have the introns, so there is nothing to be removed. Yes, non-coding sequences. Super awesome, Prachi. Okay, so over here, introns are absent in the structural genes. This is correct statement. Fine, B is correct. Now, transcription and translation are coupled process. What do you mean by that? Are they couples? Transcription and translation are coupled process means that both of them are happening together. Is it happening together in the case of bacteria? Yes. Why? Because we have studied transcription happens in the nucleus, translation happens in the cytoplasm, because ribosomes are there in the cytoplasm. In the case of eukaryotes, in the case of prokaryotes, there is no door, nothing over here, my dear children. They can easily do all basically transcription and translation together. They do not have to go anywhere else, right? So over here, this is why we say it's a coupled reaction. It can happen simultaneously. So C is also correct. Now what is D? Primary transcript undergoes splicing. So what is splicing? Splicing basically means the removal of introns is known as splicing. So over here, do you think there is need for splicing? No. So I'll say this is wrong statement. What is E statement? Only one RNA polymerase is present. Do you think only one RNA polymerase is present? Do you think only one RNA polymerase? RNA polymerase is the one that will undergo, that will help in the process of transcription. Okay, that helps in the process of transcription. From DNA, it makes the RNA. And only one is there. Yes, in prokaryotes, only one is there. In eukaryotes, there are 3 of them. Remember that. So I'll say B, C and E options are correct. So yes, you were correct when you said that. 18th answer is B. Super good. So let me read out the name of the children who actually got it correct, and by then try to solve this question. So the first person to say the answer was Prachi, then Bhagyashree, Sanika, and also Aishwarya and Manjunath. Not 18B. Okay, I think, I hope you're saying it's B only. But yes, kudos to you children. Okay, next question. RNA polymerase 2 is responsible for transcription of. Now you see, this RNA polymerase 2, whenever you have RNA polymerase 1, 2, 3, you're talking about eukaryotes. Eukaryotes are more complex organism. They're like, for transcribing this type of mRNA, I will have this. For transcribing this type of RNA, I will have something else. Okay, so what is the answer for this question? RNA polymerase 2 is responsible for the transcription of. You can be asked this question in the form of theory in your PU2 examination. What are the different type of RNA polymerases? So Bhagyashree says 19 answer is A. Okay. Aishwarya says it's C. Okay. And Manjunath says it's A. Prachi says C. Sanika says it's C. So maximum votes for C, which is why let's lock C itself. The answer is C, my dear children. So you have 3 type of RNA polymerases doing 3 things. First one, RNA polymerase 1 transcribes the rRNA. That means it forms the rRNA. What is rRNA? rRNA is something that forms the ribosome, ribosomal RNA. So you remember RNA polymerase, it starts with R, and also this guy starts with R. So the first priority is given to the one that has same letters. Okay. Now RNA polymerase 2, second priority is used to transcribe the hnRNA or basically the mRNA. What is hnRNA? So when I say that DNA is transcribed to RNA, is it directly transcribed to mRNA? No, it is transcribed to heterogeneous nuclear RNA, which you call as hnRNA. Second priority is to make hnRNA and also to transcribe and form the mRNA. Now the third priority, it starts with T, right? Third priority, so it's for tRNA, transfer RNA. Transfer RNA is there for translation process, remember? And also one type of rRNA which is known as 5S rRNA, and also small nuclear RNA. So these three things are transcribed with the help of RNA polymerase. So three things for RNA polymerase 3. Does it help you in some ways? Please remember it, it's super important. Okay, so here the answer would be C. Those of you got the answers, kudos to you. Now we are going to solve the last question, question number 20, and it is related to transcription. Histone proteins are positively charged because they are rich in basic amino acid residues. What are these residues? So histone proteins, what are histone proteins, my dear children? Basically the DNA is too long and our nucleus is too small. So to fit in, there are some proteins down there which you call as histones. Okay, so histones, they are 8 in number. 8 histones will be there. You call this as histone octamer. You call this as histone octamer. And the DNA which is there, it is wrapped around the histone octamer twice. Why do you think it is able to wrap around? So Prachi says the answer for this question is 20A. Okay, all of you actually, Bhagishree, Manjunath, Aishwarya, Sanika, all of you are saying A, and you guys are correct. Why A? Why arginine and lysine? Because, my dear children, they are positively charged. They are positively charged amino acid. Whereas your DNA which is there, it is negatively charged. Always remember it's negatively charged, which is why it is acidic also. So it is negatively charged because of phosphate group, and which is why it can come together with the positive charges. If you put negative charges in histone, it will not attach. Okay, always remember. So your answer for this question is arginine and lysine, A and L. Cool. And this is how it looks. This is structure of nucleosome. You also have one extra histone protein. You call this as H1 histone protein. And with that, we complete all the questions. Please try to solve this question. And as I said, I request you, please be there for live at a sharp 6 p.m. from the next day. Is there any challenges that you're facing for joining the live at 6 p.m. as well? Please let us know that also. So DNA replication replicates semi-conservatively was first shown in what? So this is your homework. Put it down in the comment section below. Okay, try this question and put it down in the comment section below. Fine, so that's all for today. If at all there is someone who is watching the video and have not subscribed to the channel yet, make sure subscribe to it. And so Sanika is saying no ma'am, so I think it's okay. So probably I believe that you guys will be joining from the next class onwards, and let's be consistent with that, because I'm telling you these sessions are going to be super helpful for you guys. Also in your PU2 boards. Okay? So try this question and put the answer down in the comment section. So that's all for today. Is there something else that I can help you with, or shall I end the live? Yes. Okay, cool. Thank you so much, Sanika, and thank you all the children who were there throughout. Manjunath was the first who started with. Okay, then Aishwarya Kevin, and then Prachi, Bhagyashree, I had Sanika, and everyone out there. Thank you so much. Rupa also. Okay, so bye-bye. Take care. Lots and lots of love. Wishing you all the best. You are going to absolutely rock the examination. Do not be sad about anything. Just give you 100%. Okay, so bye-bye. Take care. See you in the next live.